Human genetics has come a long way in the last 10 to 15 years. The Division of Cancer Epidemiology and Genetics, known by its initials DCEG, is one of two intramural research programs within the National Cancer Institute.
Stephen Chanik, MD, Director, Division of Cancer Epidemiology and Genetics, National Cancer Institute.
It's really focused specifically on the questions related to the etiology of cancer. So in DCEG, we study using epidemiologic and population and family studies the origins of cancer, so to speak, by looking at environmental, lifestyle, and now genetic changes and genetic variation that contributes to the risk for cancer. The Division of Cancer Epidemiology and Genetics has been a world leader for many years, really since its inception, because those who really formulated it, Dr. Joseph Fraumeni, Bob Hoover, Peggy Tucker, and others, really had this vision that conducting studies over an extended period of time and collecting biospecimens and having the best possible annotation of those specimens would allow for the future use as the technology progressed. So there was real vision 15, 20, and 30 years ago in setting up what the next generation, which I consider myself part of, have had just an extraordinary opportunity to really take advantage of this. And this in turn should pay tremendous dividends for prevention and ultimately for treatment and having a more rational strategy towards addressing the questions of cancer genetics.
Because the division has such a large investment in these population studies, over the years we've been able to genotype many samples that were part of these studies. So to date we have over 100,000 individuals here at the Division of Cancer Epidemiology and Genetics that have been genotyped.
Mitchell Makila, SCD, MPH. Earl Statman, Investigator, Division of Cancer Epidemiology and Genetics, National Cancer Institute.
And because we have such a large collection of samples, we're able to do really interesting studies with that.
Some of our studies are focused on trying to really understand the different components that contribute to a disease such as cervical cancer, which is the third leading cause of death in women in the world.
Here we designed the HPV Genomics project and developed the first high-throughput next-generation sequencing-based HPV whole genome sequencing assay.
Lisa Mirabella, PhD, MS. Earl Statman, Investigator, Division of Cancer Epidemiology and Genetics, National Cancer Institute.
Which will enable us to thoroughly study the genetics of HPV carcinogenicity. HPV-16, for example, is the most carcinogenic HPV type. It causes about half of those cervical cancers each year. So it's one of the most potent carcinogens, so it's a great tumor model. So if we understand how HPV-16 causes cancer, which we don't at this point know, we can understand a lot about viral carcinogenesis in general, which could apply to not only HPV, but other carcinogenic viruses.
We're known for playing a large role in genome-wide association studies. So that's scanning the genome of individuals using these commercially available genotyping arrays and looking for differences in inherited variation between cancer cases and a set of controls we're looking at. While we were performing these studies, we noticed that some of the samples tended to fall out with QC. And upon further investigation of that, we noticed that applying these new techniques, we could essentially create these virtual karyotypes with these genotyping arrays of the samples. And with these virtual karyotypes, we could tell that some individuals had these really large-scale deletions or gains in their genomes. And that is what we call the presence of genetic mosaicisms that we've been able to detect with these arrays and use those for further studies of hematologic cancers.
Size really does matter in genetics. Really having large data sets and having really well-curated data sets gives us an opportunity to ask questions that we never could have thought of 15 years ago. Rare diseases are a critical part of our portfolio, particularly cancer that clusters in families.
Li-Fraumeni syndrome is a genetic syndrome, and my P53 gene which is a kind of tumor suppressor gene, doesn't work properly.
Mary Fisher, sitting next to Mason Fisher, patient. Division of Cancer Epidemiology and Genetics, National Cancer Institute.
The Li-Fraumeni study started in 1969.
Maria Isabel Aschatz, MD, PhD, Principal Investigator and Clinical Geneticist, DCEG, National Cancer Institute.
And Dr. Fraumeni and Dr. Li got together and studied a large number of patients who had sarcomas at an early age, and they were able to find many family members with other types of cancer, mostly the mothers who had breast cancers. And this was the beginning of a very important finding, which was the description of Li-Fraumeni syndrome. We're now seeing around 150 families who have the diagnosis of this syndrome.
My family got tested, my children as well, and they were both diagnosed with Li-Fraumeni syndrome as well. National Institute of Health offered a study that I found out about and went ahead and applied, and our family thankfully was accepted to the study, and every year now we get our screening done here, and we're very grateful for it.
We know also that TP53 gene can be mutated somatically in almost all cancers, all human cancers. So I think that the better understanding of what happens in our families will also help us understand carcinogenesis in general.
Trainees are a critical part of all of the research we do here in the clinical genetics branch and in the Division of Cancer Epidemiology and Genetics.
Sharon Savage, MD, Chief of Clinical Genetics Branch, Division of Cancer Epidemiology and Genetics, National Cancer Institute.
We have pre-doctoral fellows, graduate students, postdoctoral fellows, many of whom have a clinical focus and others who have genetics, bioinformatics, and a wide range of other skills.
I came here as a trainee with a PhD in population genetics and infectious disease, and I came here to train to be more of a genetic epidemiologist, and I got great training while I was here and was able to transition to a genetic epidemiologist from, I think, a wet lab geneticist, and now I have 3 trainees of my own, which I'm training also to now be the next generation of genetic epidemiologists.
Genetics is really a very exciting field, but it has to be married very closely to the study of epidemiologic risk factors, because I like to think of your genome as sort of your carburetor set in a certain way, but you put your foot on the accelerator or on the brake based on kinds of decisions that you make. What happens in your life and your lifestyle and things like that affects or is affected as well what your germline does or doesn't do. So that study is just getting started. I mean, that kind of integrated study, and we see that as central to the next decade of really being able to apply our way through important questions in cancer genetics.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4CANCER. Produced October 2017.